Hello everyone, welcome to Genius Blossom. Have you ever wondered what living things are really made of? In 1665, a scientist named Robert Hooke, he looked at a slice of cork under a simple microscope and he observed tiny compartments which he called cells. Cell, it is a Latin word for little rooms. And cork, we know that cork, it is a substance which comes from the bark of a tree. So in 1665, Robert Hooke, he first observed and discovered cells from a cork tissue using a simple microscope. And that small discovery, it marked the beginning of a new chapter or a new branch in biology, which is known as cell biology. Cell biology, it is a branch of biology which deals with the study of cells. Now let us begin our fifth lesson of CBSE class 9 science, the fundamental unit of life. And welcome to the first part of our new chapter. And in this part, we will find out what makes up living things or what a cell is made of and how is the inside of a cell is arranged. Now we know that Robert Hooke discovered cells. That is, Robert Hooke in 1665 first observed and discovered cells. And cell, it is a Latin word and its meaning is little rooms or tiny compartments. And let us find out what the cells or what they actually look like. Not in a cork this time, but in something we see every day, an onion. Let us have a look at the first activity from your textbook and prepare a slide of an onion peel to observe under a microscope. And this simple experiment helps us see the real structure of living cells. If we prepare temporary mounds or slides of onion peels from different size to onions, what do we notice? Even though the onions are of different sizes, the structures we see under the microscope or we observe under a microscope, they look very similar, and these small repeating units, they make the whole onion bulb, and these structures are called, these small repeating units are called cells. These are the cells. It is not just onions. Every living organisms around us, whether it is a plant or animal or even a tiny microbe, is made up of what? Made up of cells. That is, cells are the building blocks of all life, or cell is the basic unit of life. You know that some organisms, they are made up of many cells like plants, animals, etc. While some, they live as just single cell, example certain microbes. But how did we discover all this? Let us take a quick look at the history of cell discovery. In 1665, Robert Hooke, he observed cells in a cork using a simple microscope, but the cells observed by him were dead cells. In 1674, Leeuwenhoek, he observed the free living cells in pond water. 1831, Robert Brown, not Robert Hooke, Robert Brown, he discovered nucleus in the cell. So, who discovered nucleus for the first time? Robert Brown discovered nucleus and Robert Hooke discovered cells. 1839, Purkinje, he gave the name protoplasm to the living fluid portion of the cell. In 1838, Schleiden, and in 1839, Schwann, these scientists, Schleiden and Schwann, they proposed cell theory. And what is cell theory? According to them, all plants and animals are made of cells, and cell is the basic unit of life. This is the cell theory, and this cell theory is proposed by Schleiden and Schwann in 1838 and 39. And in 1855, another scientist, Virchow, he added one more point to the cell theory, or he further expanded cell theory by adding another point, that is, all cells come from pre-existing cells. According to Virchow, all cells come from pre-existing cells, or all cells arise from the pre-existing cells. So now cell theory, it contains 3 points. All plants and animals are made of cells. Cell is the basic unit of life. All cells come from the pre-existing cells. In 1940, electron microscope was invented, helping scientists to see the very small structure or the tiniest structure inside the cell. The invention of magnifying lenses opened up a whole new world, the microscopic world. So who discovered cell? Robert Hooke discovered cell. And who discovered nucleus? Robert Brown discovered nucleus. Who coined the term protoplasm? Purkinje coined the term protoplasm. Who proposed cell theory? Schleiden and Shen proposed cell theory, and Virchow further expanded cell theory. And in 1940, what was invented? Electron microscope was invented, or invention of electron microscope. It was in 1940. Scientists discovered that some organisms like amoeba, paramecium, chlamydomonas, bacteria, etc., they are made up of just one cell or single cell. Such organisms are called unicellular organisms. Uni means one or single. So, the organisms with one cell or single cell, they are called one-celled organism or unicellular organisms. Example, amoeba, chlamydomonas, some bacteria, paramecium, etc. On the other hand, organisms like or living things like plants, animals, fungi, they are made up of many cells or more than one cells, or they are made up of multi-cells, or they have many cells working together. And such organisms made up of multi-cells or many cells, they are termed as multicellular organisms. Multi means many, many-celled organisms or multicellular organisms. Example, plants, animals, fungi, etc. Do you know every multicellular organisms, including you, starts from just a single cell? That single cell, it keeps dividing and forming more cells, creating all the body parts. So remember, all cells come from or arise from the pre-existing cells by cell division. In our second activity, prepare temporary mounds or slides, not just from the onion peels, but also from leaf peels, root tip of onion peels from different, or even onion peels from different sized onions. After observing these under a microscope, ask yourself, do all the cells, they look the same in size and shape? Do all cells have the same structure, or are there any differences between cells from different plant parts? Can we also find any similarities among them? By comparing different cells, we begin to understand how structure and function can vary even within a same plant, and how all of them still work together as a single organism. Do you know a single organism like human? It has many cells like a nerve cell, fat cell, muscle cell, blood cells, sperm, ovum, etc. And all are different in size, shape and function. All cells are different in size, shape and function. And the size and shape of each cell depends on the job it has to do. For example, nerve cells, they are long and branched to carry messages. Red blood cells, they are round and flexible to flow easily. Fat cells, they store energy and look like a small balloon. Some cells like amoeba, they have changing shapes, but many cells in our body, they have fixed and special shapes suited for their role. That is, each cell has its own division of labor. So inside every living cell, there are tiny parts called organelles, and each organelle has a special function. So remember, every cell is like a small factory with different parts and doing different jobs to keep it alive and working. Who discovered cells and how? Robert Hooke discovered cells in 1665 while observing a thin slide of cork through a simple microscope. He saw tiny box-like structures or tiny compartments and named them cells. Why is the cell called the structural and functional unit of life? The cell is called the structural and functional unit of life because it forms the basic structure of all living organisms and carries out essential life processes like respiration, growth, excretion, reproduction, etc. Our next topic is, what is a cell made up of? What is the structural organization of the cell? So let us study more details about the cell and how it is organized. We all know that we cannot see the cell with our naked eye. Then if we observe a cell under a microscope, then we would come across three features in almost every cell, whether it is an animal cell or plant cell. There are three main common features or common parts, and they are plasma membrane, nucleus and cytoplasm. Plasma membrane, it is the outermost boundary or outermost layer in the case of animal cell, but in plant cell it is seen just inner to the cell wall. In plant cell, the outermost layer or boundary is the cell wall, and inner to cell wall or just beneath the cell wall you can see plasma membrane in the case of plant cells. And nucleus, it is the controlling center of the cell, and it is found in the central part of the cell as a round or oval shaped structure, and the region in between plasma membrane and nucleus, it is called a cytoplasm. It is a fluid like structure and it is called a cytoplasm, and you can see several tiny cell organelles in the cytoplasm. Now let us check the important features of plasma membrane. Plasma membrane, it is also known as cell membrane. The other name for plasma membrane is cell membrane, and it is the outermost covering in animal cells. In animal cells, plasma membrane, it is the outermost covering, but in plant cells it is seen just inner to cell wall. So remember the point, it is the outermost covering in animal cells, and it separates cell contents or the contents of the cell from its external environment or from its surrounding medium. And the plasma membrane, it allows or permits the exit or entry of some substances through it, while it prevents the movement of certain other substances. That means it does not allow the movement of all the substances in the medium or inside the cell through it. That is, it allows only the movement of certain selected substances through it. So we can say that plasma membrane, it is a selectively permeable membrane. Why plasma membrane is called a selectively permeable membrane? Plasma membrane is called a selectively permeable membrane because it allows or permits the entry or exit of selected substances through it. That is, it does not allow the movement of all the substances through it, only selected substances can pass through it. So we can say that plasma membrane is a selectively permeable membrane. Now we have studied that plasma membrane allows the movement of only certain substances through it. But do you know how does this movement actually happen? How do substances move into the cell and how do they move out of the cell? And let us understand the different processes involved in the movement of substances across the plasma membrane. We have already studied the process of diffusion in our earlier chapters, and it is the same process, diffusion, that helps certain substances like gases to move through the plasma membrane. For example, some gases like carbon dioxide, oxygen, etc., they can move through the plasma membrane by diffusion. Then what is diffusion? Diffusion is the movement of substances or particles or molecules from a region of their higher concentration to a region of their lower concentration. That is diffusion. And here the gases like carbon dioxide and oxygen, they move across the plasma membrane by using the principle of diffusion. Some substance like carbon dioxide accumulates in higher concentration inside the cell because they are the cellular waste products. Since it is a waste product, it has to be eliminated or removed from the cell. And the concentration, since the concentration of carbon dioxide is higher inside the cell than the surrounding medium, the process of diffusion takes place here. Then the process of diffusion, what is diffusion? Diffusion means the movement of particles or substance from a region of its higher concentration to a region of its lower concentration. Here the concentration of carbon dioxide is higher inside the cell, then it moves towards the outer medium, because in the outer medium the concentration of carbon dioxide is lower compared to the concentration of carbon dioxide inside the cell. So here the movement of carbon dioxide from the cell towards the surrounding medium takes place, and it occurs through the plasma membrane, and then it occurs by the process of diffusion. Similarly, oxygen also enters the cell through a process of diffusion when the concentration of oxygen inside the cell is lower than that in the outside or in the surrounding medium. And thus the process of diffusion helps in the exchange of gases. Water also follows the rule of diffusion, that is, it moves from its region of higher concentration to the region of lower concentration, and when such type of movement happens with the gases, we call it as diffusion. However, if the movement involves water through the semi-permeable membrane, we do not call it diffusion, but we use a special term for it, and that is osmosis. So osmosis is the movement of water from its region of higher concentration to the region of lower concentration through a semi-permeable membrane or through the selectively permeable membrane or through the plasma membrane. So, during osmosis, only water molecules move through the plasma membrane, and it occurs through a semi-permeable membrane or plasma membrane, and it follows the principle of diffusion. Osmosis follows the principle of diffusion, but in it only the movement of which type of molecules occur? Water molecules occur. Also, the movement of water across the plasma membrane, it depends on the amount of substance. Substance means salt, sugar, etc. The amount depends on the amount of substance that is dissolved in the water. Osmosis, it is the movement of water through the selectively permeable membrane or plasma membrane from an area of more water. Area of more water means there the dissolved substance like salt or sugar, its amount is in very low quantity, that is less dissolved substance. More water means less dissolved substance, to an area of less water. Less water means there more dissolved substance. The amount of sugar or salt is more there. This means that water moves towards the side with a higher solute concentration. We can say that osmosis, it is the net diffusion of water across a selectively permeable membrane toward a higher solute concentration. What will happen if we put an animal cell or plant cell into a solution of sugar or salt in water? Now let us understand what happens when a cell is placed in different solutions, different types of solutions. And here the key factor is the concentration of water outside the cell compared to the concentration of water inside. And based on the concentration of water in the surrounding medium, water may enter the cell, leave the cell, or move in and out equally. And this gives us three types of solutions, that is hypotonic solution, isotonic solution, and hypertonic solution. In hypotonic solution, the water concentration in the surrounding medium, not inside the cell, the concentration of water in the external environment or the surrounding medium or outside the cell, it is higher water concentration. If the water concentration is higher in the surrounding medium, then what happens? Water enter into the cell, because the water inside the cell is low compared to the water outside the cell. So water enters into the cell, and as a result the cell swells. And such type of solution is called a hypotonic solution. That is, the concentration of water in the surrounding medium is higher than that inside the cell. And if the cell placed in isotonic solution, isotonic means there the concentration of water in the surrounding medium and inside the cell both are same, both the concentrations are same. The concentration of water inside the cell and in the surrounding medium, when they are in the same concentration or equal concentration, then the water moves in and out equally. As a result, no net movement of water, and such type of water, that is the concentration of water in the surrounding medium and inside the cell, they are same. And such type of solution is called a isotonic solution, and here the cell shows no change because water moves in and out equally. The concentration of water in the surrounding medium is lower than that inside the cell, then what happens? Then water moves towards the surrounding medium, or water moves out of the cell into the surrounding medium, and in that case the cell shrinks. As the water enters into the surrounding medium or water moves out of the cell, then the cell shrinks, and such type of solution is called a hypertonic solution. So we classify solutions into hypotonic solution, isotonic solution, and hypertonic solution based on what? Based on how water moves through the plasma membrane. Now let me describe an activity involving an egg that clearly demonstrates the process of osmosis. Take an egg and remove its shell by placing it in the dilute hydrochloric acid, and the shell dissolve, leaving a thin membrane. Now place this egg in pure water and observe after 5 minutes. After 5 minutes, what happens? The egg swells because the concentration of water is outside in the surrounding medium, and from the surrounding medium the water enters into the egg, and as a result the egg swells. But if we place the same egg in the concentrated solution and observe after 5 minutes, then you will notice that the egg shrinks. Instead of swelling up, the egg shrinks. Why? Because in the concentrated salt solution, the amount of water or the concentration of water is very much lower or lesser than the inside of the egg. So from the inside of the egg cell, the water moves out into the concentrated salt solution. You can also understand the concept of osmosis with the help of a simple example using dried raisins. In dried raisins, we know that the concentration of water is very low. When we put it in pure water, it absorbs water by the process of osmosis. That is, in pure water, the concentration of water is very high, and in raisins, water concentration is very low. So, from pure water, the water particles move towards the raisin cells, and as a result, the raisin swells up. And this swelled resin, if we put it in the sugar solution or salt solution, then what happens? Inside the swelled resin, the concentration of water is very high. In salt or sugar solution, water concentration is very low. Then the opposite process of this happens in the salt solution. That is, the water inside the raisins, swelled raisins, they move out of the cell into the surrounding medium or into the salt solution. That is, the by the process of osmosis, the water inside the raisins move into the salt or sugar solution. Unicellular freshwater organisms and most plant cells, they usually gain water through the process of osmosis. A very common example of osmosis in everyday life, it is the absorption of water by plant roots. And this process occurs very naturally because there the water moves from soil to the plant cells. In the soil, concentration of water is very high, and in plant cell, concentration of water is low. So, movement of water occurs from the soil to the plant cells by the process of osmosis. Diffusion plays an important role in the exchange of gases and water in the life of the cell. And along with this, the cell, it needs some nutrients, and it takes that nutrients from its surrounding medium. And for the movement of nutrients in and out of the cell, they need energy. And the movement of nutrients using energy, it is called active transport. That type of movement is called active transport. The plasma membrane, it is flexible. Plasma membrane is flexible, and it is made up of organic molecules like lipids and proteins. But its detailed structure, it can be seen only under an electron microscope. We studied that plasma membrane is flexible, and that flexibility of plasma membrane, it helps the cell or allows the cell to engulf or take in food and other materials from the surrounding medium. And that process is called endocytosis. The process of taking food or engulfing food from the surrounding medium using the flexible plasma membrane is called the endocytosis. And amoeba, example amoeba, it captures its food from the surrounding medium by using this method, endocytosis. Now it's time for summary. Here is a quick summary of what we have discussed in this topic. The cell and its discovery, history of cell discovery, unicellular and multicellular organisms, structural organization of the cell, three main features of the cell, plasma membrane, nucleus and cytoplasm, diffusion and its role in the movement of gases in cell through plasma membrane, osmosis and movement of water through plasma membrane. I hope the concept is now clear to you. Thank you for watching.